Hello there. Welcome to DNA Edits. In today's lecture, will describe the what is transcription, what is reverse transcription. In addition to the description, we will also dissect the differences between the transcription and reverse transcription. So first the transcription. Transcription is where RNA is synthesized from the DNA. RNA is synthesized from DNA is called as transcription. In reverse transcription, DNA is synthesized from RNA. DNA is synthesized from RNA. So transcription is very common in prokaryotes such as bacteria and eukaryotes. And reverse transcription you can see in retroviruses. Even coronavirus, which belongs to retrovirus, where reverse transcription takes place. Okay, so I have drawn the table where 10 to 12 differences are there between the transcription and reverse transcription that we will check within a second. This table describes the differences between the transcription and reverse transcription. The left side have written features, and the center one is transcription, and the right one is a reverse transcription. First, we will see what is that enzyme and the subunits required for the transcription and reverse transcription. In case of transcription, there is an enzyme called as RNA polymerase. E. coli and eukaryotes, a family of enzymes called as RNA polymerases, they catalyzes the transcription, nothing but synthesis of RNA from DNA. And you see E. coli RNA polymerases are having 5 subunits, whereas eukaryotic RNA polymerases are having 5 to 12 subunits. In reverse transcription, there is an enzyme called as reverse transcriptase. The enzyme called as reverse transcriptase is a heterodimer. In the reverse transcription, RNA is converted to DNA molecule. So this is carried out by the enzyme called as reverse transcriptase. Does this RNA polymerase or reverse transcriptase, do they have any other activities like other activities? RNA polymerase do not have any proofreading activity. And when it comes to the reverse transcriptase, it is having a special activity called as RNase H. H stands for hybrid. Whenever RNA is converted to DNA, it forms the double strand hybrid. One strand is mRNA, other strand is DNA. So this is a hybrid molecule. So because of RNase activity, the RNA molecule is degraded. There is one more activity that is DNA polymerase. That DNA polymerase is useful in the conversion of single strand DNA to double strand DNA. So reverse transcription requires the reverse transcriptase and RNase H activity and DNA polymerase. Whereas transcription is required only one enzyme, one activity, that is RNA polymerase. That's a conversion of DNA to RNA. So here the third feature is template. What is the template here? In case of transcription, it is a DNA. In transcription, template is DNA from which RNA is synthesized. In reverse transcription, RNA is template from which DNA is synthesized. The next one is primer. Transcription do not require any primer. There is no requirement of primer in the process of transcription. But in case of reverse transcription, there is a primer requirement. In the reverse transcription, in the natural process, host tRNA acts as a primer. Host tRNA acts as a primer to synthesize the first strand of DNA. Any other proteins are required for the transcription? Yes. Transcription factors helps in the transcription where they'll bind to the promoter region and they'll speed up the transcription. In case of reverse transcription, there are molecules called as nucleocapsids. Nucleocapsids, they are required for the reverse transcription. What is a product in transcription? RNA is a product. In reverse transcription, DNA is a product. Yeah, the location of synthesis is the most important. Transcription takes place in the nucleus. Transcription takes place in the nucleus, whereas reverse transcription takes place in the cytoplasm. Where it is? Cytoplasm. And the synthesized DNA is transported to the cytoplasm. Target location is a cytoplasm, where the DNA synthesized in the process of reverse transcription is target location is nucleus. So what will happen to the fate of the molecule? Translated mRNA is converted into protein in transcription, whereas in reverse transcription, the DNA which is synthesized is transported to the nucleus, where it is integrated into the host chromosome. It is integrated into the host chromosome. In transcription, RNA is converted to mRNA converted to protein, and in the reverse transcription, it goes and integrated into the host chromosome. So if you see what are the inhibitors of transcription in E. coli or prokaryotes, it's a rifampicin. In eukaryotes, it's a alpha amyloidin is used as a inhibitor of RNA polymerases. When it comes to the reverse transcriptase, there is a molecule called as azidothiamidine is used as a inhibitor of reverse transcription. It blocks the conversion of RNA to DNA molecule. So this is a part of antiviral treatment, retroviral treatment. So AIDS patients are given this drug called as azidothiamidine, and there is another molecule called as a nevirapine. This molecule is used as again, this inhibits the reverse transcriptase. So azidothiamidine inhibits the reverse transcription by blocking the chain elongation, whereas nevirapine, this molecule inhibits the reverse transcriptase enzyme itself. If you see the speed of RNA polymerase in a transcription, this is around 40 to 80 nucleotides per second. 40 to 80 nucleotides per second. But when it comes to the reverse transcriptase, this is 15 nucleotides per second. And people say that reverse transcriptase is a sluggish enzyme. Sluggish enzyme. Why? Because when virus infects the cell, from infection to the first appearance of DNA molecule, it takes 4 hours. For 9 kb molecule, to synthesize the 9 kb, it taking 4 hours time. So probably the name that given to this enzyme sluggishness is really true with the reverse transcriptase. It is just 15 nucleotides per second. It takes 4 hours time to see appearance of DNA molecule after infection. And the errors are mutations, and it is almost like 10 to the power of 4 or 10 to the power of 5, means 10,000 nucleotide per one error or one mutation in 1000, 10,000 nucleotides roughly. But here you see one in every 1500 to 400, 1500 to 4000 nucleotides. So you see, this is what is the advantage to the retroviruses, where their DNA is, their genetic materials keep on changing because their reverse transcriptase is not having any proofreading activity. It entertains the uh, variations, and that leads to the loss of its viability or adapting to the new environment. So that is there. And the nucleotides, transcription requires the ribonucleotides, and the last one deoxynucleotide. I hope this differences between the transcription and reverse transcription will help you to understand they really understand the differences, right? uh, if at all you like it, just keep watching for my channel for regular updates. Thank you all.